Welcome everyone to the CLIMOS webinar series. My name is Edwin Kniha, and together with my colleague Katharina Platzgummer, it is a really great pleasure to dive into the topic of sandflies in Central Europe today. To the program, we will provide you with a general short overview on the topic and go a little bit into detail on Central Europe. And we will also show you a bit of applied research and what our role is in CLIMOS and what our everyday life looks like. Let's start very generally. The broad topic behind that today is vector-borne diseases, which are, well, by definition, illnesses caused by viruses, bacteria, and parasites that are transmitted by so-called vectors. In our case, the vectors are the sandflies. The pathogens that we are looking at in detail and that can be transmitted are, first of all, Leishmania and flevoviruses. And if we look at a third very important part of the transmission cycle, we have to look at the reservoir species as well. And those are, well, many different vertebrate species, and I will show you a little bit more in detail what species we are talking about when we look at the sandflies and sandfly-borne diseases. First of all, I would like to guide you a little bit through, probably the most important thing about it, the vectors, the sandflies. Approximately 950, even a little bit more species have been described from all around the world, from tropical, subtropical, arid, temperate regions. They are dipteran insects, so they have two wings, and they belong to the family Psychodidae, the moth flies, so they are the subfamily Phlebotominae. You can see two beautiful specimens here on the picture. They have some quite distinctive features where you can morphologically discriminate them from other species or insects. They are very small. They keep their wings in this erect V-like shape whenever they are feeding or resting. The name sandflies actually comes from their color, which is usually, well, golden, grayish, sandy color. And as you can see on the picture here, the body is densely packed with what you would call hair-like structures, and the legs are quite long and delicate compared to their body. Very importantly, both sexes of sandflies feed on sugar sources, and only the females bite vertebrate hosts. So only the females are blood feeding, which is of course also crucial for transmission cycles. So only the females are involved in the active transmission. Usually they are most active during dusk and dawn, and when they approach you, unlike mosquitoes, for example, they are silently approaching, so you don't hear them at all when they come to feed on you. And the feeding process, so the biting, is sort of described as painful. Of course, not excruciating, but compared to other blood feeding insects, you would probably feel a bit of the bite. And this usually can result in redness and itchiness, and particularly if you have a lot of bites, so after nights of heavy biting, there can be quite persistent redness and swelling, even secondary infections. And particularly if a pathogen has been transmitted, you would look out for symptoms such as fevers or chills, and in case of Leishmania, also of non-healing sandfly bites that would eventually develop into sores and ulcers or larger lesions. Discriminating a sandfly from other, particularly small dipteran insects, can be quite challenging, and it's not very easy. Therefore, I would like to draw your attention quickly to a very nice guidance and brochure that was set up in the frame of the CLIMOS project that would, in case you are interested, help you to discriminate and identify sandflies from other insects, and also in addition guide you through the best protection against the bites, and it is available in many different languages, and you can access it online, and it is really a great tool. Looking at the gonotrophic cycle, it starts with two adult specimens, a female and a male, that are mating, and the female needs to take a blood meal in order for the eggs to develop. And then the cycle continues in soil, so it is strictly terrestrial. You can see that here. Females are depositing their eggs in soil, and from those larvae would emerge in 4 instars, molt into pupae, and the pupae are then emerging again as adults altogether, taking a couple of weeks, of course, very much dependent on the temperature. And particularly these pre-adult life stages happening in soil make it quite challenging for us to detect sandflies or to detect particularly pre-adult stages, as they are very well hidden. And of course, this terrestrial life cycle somehow also influences the habitats where sandflies would be present, that are actually very diverse. If you look at just some of them, it can be very rural, including forests, for example. Some species would breed in rodent and animal burrows. But on the other hand, and also very often in Europe, we would find sandflies very close to human habitats, to human dwellings, where you would also find animal hosts, like animal farms, old barns and sheds, old cellars of houses, even rock walls and crevices, and also, for example, caves. So you can see that habitats can be very diverse wherever you look at. Coming to the pathogens involved, most importantly for sure, the protozoan parasites Leishmania that belong to the Trypanosomatidae family. The life cycle, in very brief, an infected sandfly will transmit so-called promastigote forms to very different vertebrate hosts, you can see, including humans, dogs, and many others, and then take up another blood meal from an infected host, and those so-called amastigote forms are then transmitted to the sandfly again, and the cycle closes. Approximately 98 countries or territories are endemic for leishmaniasis, and the WHO estimates that approximately 1 billion people are actually at risk of infection. And not going into detail, because leishmaniasis is a very complex disease. The visceral form, the deadly form in humans, causes approximately 50 to 90,000 new cases, and is, if untreated, most likely deadly. And the cutaneous, the second major form, the cutaneous leishmaniasis, approximately causes 1 million cases annually. And if you look at the clinical picture, you would see that here on the left side, you see a so-called an ulcer or a sore of a cutaneous case of human leishmaniasis. But at the same time, you see a lot of pictures from dogs, which are actually the main reservoir hosts for Leishmania infantum, the predominant Leishmania species transmitted in Europe. So also very important for us here. The dogs are reservoirs. They are important hosts. They can get sick, as you see here on the pictures, and the disease usually, if symptomatic, is systemic and can affect multiple organs and often results in death and a lot of suffering in the dogs. On the other hand, we have the fleboviruses that are mostly transmitted by sandflies. In Europe here exclusively, we have sandfly fever, Sicilian virus as well, one of the most important ones transmitted. You can see here on the map the distribution, or the known distribution, colored. Most of the infections are asymptomatic. If symptomatic, they will result in influenza-like syndromes that would usually be self-limiting. And we have, on the other hand, Toscana virus, and also here you can see autochthonous and important cases mapped on the map here. The difference is that Toscana virus is neurotropic and can also cause acute meningitis and meningoencephalitis, and thus is of high medical relevance. Let's quickly look at the reservoirs. If we draw our attention to Europe and Mediterranean parts of Europe, the dogs are for sure the most important reservoir species, as Leishmanian phantom said before, is the predominant species and mostly affecting dogs. But as part of a zoonotic transmission cycle, also humans can be involved in the transmission, and Leishmanian phantom can, of course, be transmitted to humans. If you look at many other host species, Leishmanian phantom DNA has been detected from cats, from rodents, horses, and many other species, but the dogs remain the most important reservoir species here. Yeah, let's go a little bit into detail on the situation in Central Europe. And if I talk about Central Europe, I am referring mostly today to Austria, to Germany, the Czech Republic, Slovakia, Hungary, so all parts of Europe around Austria that seem to be of importance for us and for sandfly-borne diseases. The interesting thing about Central European sandfly distribution is that for a long time, the common opinion was that sandflies are not present north of the Alps, and only in 1999, so not that long ago, the first sandflies were detected in Germany, and yeah, a couple of years after in Austria in 2008, and even later in Slovakia in 2016, and also most of the records in Hungary, for example, are quite recent records with some very few historical ones, but apart from those, most of them are very recent records. And I would like to draw your attention to this distribution map here. And you see as colored dots different sandfly species that have been mapped here. Well, it can be seen very quickly that Phlebotomus marciti here, colored in red, is the predominant species in Central European countries. You see them here present in the so-called Rhine Valley in Germany, in all warm parts of Austria, but also in the bordering countries, Hungary, a single record in Slovakia, even up to Belgium and Switzerland. And then there are additionally some quite peculiar records of, for example, Phlebotomus perniciosus here in yellow in Germany and Phlebotomus simitzi in Austria. Both of them have only been recorded sporadically, and by that I mean only one time and then never again. So this, of course, gives rise to a lot of different questions. Some of those cannot be answered at the moment, but I will go a little bit more in detail on that. And if you have a closer look on the map, you will see that, for example, in warm parts of Hungary, we have even more species, and also going more south, here in Italy and Slovenia, we have more species present. But the Central European sandfly fauna is very restricted, and in principle, only one species is predominantly present. And after the first records, the opinion was that probably climate change was the main driver of sandfly emergence in Central Europe. However, it became rather evident, and we ourselves did a few studies on that actually sandflies emerged after the last glacial period, already in the last, well, couple of hundred or couple of thousand years already, and have just been overlooked for a very long time. And still not fully clear, but probably sandflies used various routes of dispersal. So probably some populations came from a refugial area here in France, and some others, like Phlebotomus simici, came from a refugial area in the Balkans. But this is yet not fully clear, because also we don't have a very good picture on their dispersal ability. So we are still not fully sure how fast and how sandflies are actually dispersing. And it becomes quite clear that the sandfly distribution in Central Europe surely is yet underreported. We have done a lot of surveys, but still there are a lot of areas that have been unsampled to date. As I said before, the detection is challenging, so you would only find adults flying around, and also at some places, or at many places and areas, sandfly abundance is not very high, and you don't find too many specimens. And moreover, if you look at the trapping success, then we approximately only see 1 out of 10 sampled sites positive, which makes it very, very hard to detect, because we have to sample a lot of sampling sites in order to find a positive one. And in addition, we somehow apply a sampling bias. We usually focus on sites close to humans and animals. I give you an example here. This old barn, very close to human settlements, is a perfect candidate, and was one of the sandfly sites that we found in Styria, and those represent usually the ones that we are looking at. But we somehow neglected and still neglect more rural areas. As you can see here, this is a quarry in Hungary that we have recently sampled, but also here, as we saw, for example, from Hungary, we found sandflies there. So they are also present in very rural areas, which might even provide stepping stones in between. And we should probably also focus more on those areas in order to get a better picture on the actual distribution. And surely temperature is a driver for dispersal. We see that there is recent northward spread. There are some or few studies in Italy or also Switzerland that report a northward spread of some sandfly species, most likely due to a rise of temperature. For that, of course, it is very crucial to have baseline data, historical data that you can compare with current data, and the baseline data is the data that we are collecting today and tomorrow, and in the recent past and future, in order to follow potential spread of the sandflies in the next years. And also here I'm providing you another map, and you can see again colored the different sandfly species, but what you can also see is as different grayish symbols here, suspected or even confirmed autochthonous cases of leishmaniasis canine, human, bovine, and equine cases. And you see that those cases are rather rare in Germany, in Austria, in Hungary, and in Switzerland. Why? Because there are probably not as many cases as in other endemic countries, and surely Austria and Germany are not considered endemic countries for leishmaniasis yet, but also because leishmaniasis is not a notifiable disease, not in humans and not in animals, and we only have a few studies on the prevalence and the seroprevalence of leishmania in humans and animals. And a second important thing about that is that we still don't know whether Phlebotomus marciti, the predominant sandfly species, is actually a vector of leishmania or not. There are some studies that reported the detection of leishmania infantum DNA in Phlebotomus marciti, and we know that Phlebotomus marciti is a multi-host feeding species, which would maybe contribute to a transmission between host animals and humans, but still the vector capacity has not been clarified, and we don't know whether Phlebotomus marciti can transmit or not. And flebovirus circulation is really a blank space here. It is, well, yeah, unknown. There are only a few suspected autochthonous transmission cases in Germany, but apart from that, we don't have any evidence of flebovirus circulation in Central Europe. And by that, I would like to hand over to my colleague who will give you another very interesting information.
Yes, very good morning also from my side. Thank you, Edwin, for this nice introduction. I think it became very clear that we already know a lot about sandflies, although they have been found only recently in Central Europe, but I think there are a lot of questions still to answer, and that's why it's very good that we are involved also in the CLIMOS project, because that is going to help us with that. CLIMOS stands for Climate Monitoring and Decision Support Framework for Sandfly-borne Diseases Detection and Mitigation with Cost-Benefit and Climate Policy Measures. And a lot of partners are involved in this huge project. So you can see here on the right side shaded in blue that we are distributed not only in Europe, but also beyond. 29 partners overall under the leadership of our great coordinator, Carla Maia from Portugal. And all of us are working on 7 different work packages. The Medical University of Vienna, so us, are involved mainly in work package 2, namely sample collection and processing. So that's why I'm going to tell you mainly about that today. However, if you want to know more about the CLIMOS project in general, I invite you to have a look at the first webinar in our series, where you get a nice introduction into sandflies, sandfly-borne diseases, and of course also the CLIMOS project. And in general, you get a lot of information, not only on the CLIMOS project, but also on training material, interviews, or sandflies in general, if you visit the CLIMOS YouTube channel. However, let's now focus on the work package 2, which starts basically with the most important thing, the sandfly surveillance. Fieldwork on sandflies is conducted in 11 countries within the CLIMOS project. You can see them here in green, and you can see that the Medical University is also involved here. It is actually responsible for Austria and Germany. All of the partners follow a twofold approach. So first of all, we are collecting temporal data. So we want to find out more about the seasonality of sandfly activity. By that, in 2023, 86 locations have been covered all over the different countries, summing up to nearly 4,000 CDC light trap nights. So you can imagine that's a huge amount of data that is collected and then needs to be processed by our modeling colleagues. In addition, we also want to know the distribution of the sandflies in these different countries. So that's why we also collect sporadic trapping data, meaning that we are going to locations that have never been tested before to see if sandflies are there or not. In addition to the sandflies themselves, to the traps themselves, we also deploy data loggers or UV bot devices to collect microclimatic data, so humidity and temperature, UbiBot devices are not used by all of the partners. They are connected to the cloud, so they allow to collect more data. However, they are not really placed at every trap location, and we did not have them. So I'm going to show you the data loggers today. All of the partners used the same traps. It's so-called Center for Disease Control light traps. It's a battery-powered trap, so the battery powers this light bulb here. So any insect that is attracted by light will come here, will come to the light bulb, and then be sucked into a net by this fan here. And in the net, they will be trapped until we collect the net, which is usually on the next day. Here on the left, you can also see this data logger. It's basically a large USB stick that allows you to collect data on temperature and humidity really at the site of the trap, so where the sandfly is actually living. As in all the other countries, we did these regular testings to collect their temporal data. This means we deployed these traps for 2 nights each month. In Central Europe, we did this between April and October. This is because we know that between November and March, it's still too cold for the sandflies to be active, so we won't be able to trap any. However, these time periods are, of course, different for the more southern countries. In addition, we also did this testing of the new sites to collect spatial data to know where the sandflies actually are distributed. This we do for 1 to 2 nights each in July and August. We do this in July and August because we know that sandflies are most active in July and also in the warmer parts of August. So if the sandflies are present somewhere in July and August, we are really safe to also have them in our traps then. So if we have a quick look at where our regular sandfly monitoring sites are, it reflects very well how the sandflies are also distributed in Central Europe, or the knowledge that we have so far about them. We had overall 20 regularly sampled locations. In Germany, we had our colleague Sandra Oerter collect the sandflies for us. So she did a really, really terrific job at sampling these 6 locations in Baden-Württemberg and 6 locations in Rheinland-Pfalz for 2 nights each month. And by that also was able to find a lot of sandflies for us. In Austria, we also had some support by the Austrian Army that helped us to sample these 8 locations overall. 3 of them are in Styria, 1 in Burgenland, and 2 each in Vienna and Lower Austria. And also here, we were able to find sandflies again. When we are looking at these sites, it also reflects again that we are mostly looking at sites that are kind of human-made. However, we also have one site in Germany where we have this rather natural cave where bats are living in. So here the blood meal host is probably bats, but most of our sites are actually human dwellings. And here you can see that the sandflies are also very near to the host animals, which in this case are our domestic animals. And here it is very important that the sandflies are not disturbed too much so that they can also complete their life cycle in these human near habitats. If you want to know more about the other trapping sites of our other partners in the CLIMOS project, I again would say you should visit our project website where you can find out a bit more about also the other partners' trapping sites, since they of course can be looking very different than ours. Now, I already mentioned that the next day, usually we are collecting the traps and then we empty these traps. And as you can see here, I think it makes very clear why we also called this webinar Finding a Needle in the Haystack, because apart from the fact that we need to find the sandfly locations first, which is not too easy in Central Europe, we also then need, in between all these other insects, pick out the sandflies. And when we have found our sandflies in this huge haystack of other insects, we dissect them. So we want to mount the head and the abdomen on microscope slides, because these are the body parts that allow us to do morphological species identification. So here in the head, you have this structure called the pharynx, with the pharynx armature that is actually species specific, same as these appendages of the male genitalia, and here the spermatheca in the female abdomen. And when we did the morphological identification, we then are going to our molecular work. So in Austria and Germany, we are actually able to analyze every single sandfly individually. We don't have to pool them as our other partners that have much more sandflies compared to us. But this allows us to really look at every single sandfly. So with the remaining body parts, we homogenize it, and then from this homogenate, we do DNA and RNA extraction, and then we use PCR-based methodologies to do the pathogen screening, so to screen for fleboviruses and leishmania in the sandflies. And if we find something positive, we send our material to our partners from the CLIMOS project to validate what we did, to have really the confirmation from 2 independent laboratories. However, Edwin already mentioned that when we are talking about the pathogens, it's important to not only look about the pathogens in the sandflies, but also in the potential reservoir hosts. There are a lot of different hosts that are in principle susceptible for leishmania and fleboviruses, but in general in Europe, but also especially in Central Europe, it's definitely the dogs that need to be considered the most important ones that we have to have a look at. And that's what the CLIMOS project does as well. But let's first have a look at the situation as it is now. So when we look, for example, at the canine leishmaniasis seroprevalence, we see that especially in these countries such as Spain, Greece, and Portugal, where we import a lot of dogs from into Central Europe, we have quite a high seroprevalence. So for example, here in these countries, more than 1 in 5 dogs has been in contact with leishmania before. And the numbers are even higher when we look at the fleboviruses, of course, always depending on the country, but we can see that also in Spain, in parts of Italy, the numbers of Toscana virus seroprevalence are very high. And for Sicilian virus, we have also data from Cyprus, Portugal, and Greece. Again, countries where there are some dogs imported to Central Europe. So it's important to have a look at this, to know this, and also it's important to have standardized data. And that's also why the CLIMOS project wants to have a closer look at this. The general CLIMOS approach is that our colleagues from the more endemic countries, so to say, are driving to the regions where they found sandflies before and really get the blood from dogs living there and screen that for pathogens. However, this is unfortunately not possible for us in Central Europe, since we are not endemic countries for the sandfly-borne pathogens. We need to really screen a lot of dog sera, and we are simply not able to find so many dogs in the areas where we find sandflies, which is why we are very, very happy that we have a collaboration with the IDEX laboratories. They are centralized veterinary laboratories where veterinarians can send the blood of the domestic animals for blood analysis, and they were so kind as to collect a lot of dog sera for us in the last 2 seasons so that we can then screen them for leishmania and fleboviruses. How are we doing that? For the detection of Leishmania infantum, we are using an immunofluorescent antibody test against Leishmania infantum. And to screen for fleboviruses, we use seroneutralization assays against sandfly fever Sicilian virus and Toscana virus. This is done by our 2 partner laboratories that have the equipment to do that. And both of these methods are indirect methods, so it's not screened directly for the pathogens in the blood of the dogs, but it's indirect methods. So by this, we can find out if this dog has been in contact with Leishmania or fleboviruses before. Now, coming to the end of our presentation, I want to emphasize how important it is to really apply a One Health approach when it comes to vector-borne pathogens in general, but also to sandfly-borne diseases, of course. I think we made it quite clear during the presentation that not only human, but also animal health is impacted by sandfly-borne pathogens such as Leishmania and fleboviruses. So it's very important to not only look at human data, but also animal data. And of course, when it comes to vector-borne diseases, it's very important to also include environmental health in the considerations. We know that climate change will probably lead to a further distribution of different vectors and also sandflies, because the environmental conditions will be getting better and better and warmer. So it's very important, if we really want to better understand vector-borne diseases and also what we can do about the distribution, for these different disciplines to work tightly together. With this, I want to thank you very, very much today for joining. And if you want to have more updates and not miss one of the further webinars that are going to happen very soon, I would invite you to have a look at our social media channels. With this QR code here, you can access them all and also come to the CLIMOS website. And yeah, I thank you very, very much for being here with us today, for listening, but also for the discussion afterwards. We are quickly, for transparency, showing also the literature that we were using for the webinar presentation today.